So, for lab, we're doing metabolic tests right now. I think I continue that. And that these are good ways of classifying bacteria, looking at some of their very specific characteristics in terms of what do they like to have for lunch. And the phenol red tubes that we used, it uses something called phenol red base, which is a protein solution and the phenol red pH indicator and some salt. And pretty much that's it. So the protein, it's a casein hydrolysate, which is just milk protein, provides the nitrogen source and the carbon source, although having to break down proteins is not all that easy for the bacteria, and they absolutely cannot ferment the protein. Remember when we were going through metabolism, them some blue Crocs, dude. All right, when we were going through metabolism, we were talking about anaerobic respiration, which is where we're using something as the final electron acceptor that is not oxygen. And it's part of the electron transport chain. And then we talked about fermentation, which is where we are taking NADH and dropping its electron onto pyruvate and recycling it back in glycolysis. And that's fermentation. And like you and I, we make lactic acid, yeast make ethanol. Bacteria can make some phenomenally weird-ass stuff. And I just been sort of looking at it and thinking about it, and that they can make lactic acid, but they can also make acetic acid, which is a 2-carbon molecule. And so one of the carbons has to get popped off. They can probably make acetone. They can do nail polish remover. They can make all kinds of odd things when they're doing fermentation. So they're taking pyruvate, dumping the electron on it, and then doing other things with it. What this means is, when you're carrying out fermentation with bacteria, they can produce an acid that's going to take that phenol red, make it go from red to yellow. They can potentially take that acid, pop a carbon off of it, and actually generate a small amount of carbon dioxide, and that's what fills up that little Durham tube. So you can have guys that are going acidic, guys that are going acidic and kicking out a carbon dioxide. Then you have guys that, given that sugar, they just can't metabolize it that way, or the way they're metabolizing it is just not producing a change in pH. We also have some odd bacteria, I'm not sure that we're actually looking at any, that are saying to heck with sugars, and they really are breaking down a lot of the protein, and they're kicking out the ammonia from that amine group and actually making the solution more basic rather than more acidic. And it just goes to the point that bacteria are doing a bunch of different things. And it's one of the things for myself thinking about, I am such a strict organo chemoheterotroph in that, that's just the way every creature I know, my dog, my family, the bunny rabbit, operates, in that we're getting both our energy from carbon molecules and our electrons from carbon molecules, and we're using oxygen. Bacteria can do some really bizarre things. And so if you turn off the oxygen for you and I, we carry out fermentation by making lactic acid. And pretty much if you're an animal, that's all you can do. Plants are going to be producing ethanol, but that's really the range of things in the eukaryotic world that we're generating through fermentation. Bacteria, man, just the same way they can eat a lot of different things, they do all kinds of odd things with fermentation. So consequently, that sugar assay using sucrose, lactose, glucose ends up being actually a pretty good way of starting to identify individual bacteria, because we know certain bacteria are going to respond to those conditions in very specific ways. So we're going to continue on with more tests that will allow us to differentiate individual bacteria. Anyway, after class, if you haven't looked, we go upstairs and look. The starch experiment worked really well in that we got guys that were breaking down starch, and they generate a clear spot in the starch plate when you put the iodine on it. Iodine reacts with starch really strongly. And the lipase experiment, I think both of those bacteria strains we're using are lipase positive because they did a pretty good job of taking that blue dye and converting it into the colorless form, which means they're kicking out free fatty acids. And that's the main way of looking at it. Anyway, these are all different proteins, and proteins are coming from and coded for by genes, and we can transfer genes around. So we're thinking about the genetics of like antibiotic resistance. We're talking about the ability of taking DNA sequences and moving them from one organism to another. And so we've got, the standard way you move genetic information from one organism, from one cell to another, is through cell division and through propagation. We have sexual reproduction. Bacteria pretty much are doing strictly asexual reproduction. Bacteria can also carry out what's known as horizontal gene transfer, in which two unrelated guys can swap DNA, or DNA from one guy can go to another guy. And we really don't have horizontal gene transfer much. No, you can have infectious viral particles go between individuals, though, but you really don't have a way of transferring genetic material from one individual to another. And it's just one of the interesting things from a developmental biology, biotechnology perspective. A single-celled organism like bacteria, man, you can genetically modify them very easily because all you got to do is throw a piece of DNA into that cell. Bing, that whole organism is now carrying that new piece of DNA. With you and I, genetic modification can happen with individual cells, but to put a new gene into every cell of your body is pretty much impossible at this point. The only time you can do that is prior to fertilization. You can shoot a little bit of DNA into the egg, fertilize it, and while not every cell will take it up, most will, and it, we can get into that further, but in terms of making recombinant complex organisms, it is not easy to do. It's one of the things like with gene therapy. People that have cystic fibrosis are missing the chloride transporter in their lungs. And it's very difficult to try to treat that. Because it's in their lungs, one of the things you can do is give them an inhaler that is actually carrying a little bit of virus, or probably now RNA would work. You puff that, and you can actually add the chloride transporter back to their lung cells. The problem is that it's not going to be maintained because you're not going to get into the cells that are, as we think of it as the stem cells that are generating the lung tissue. You're just getting it into the lung epithelium, and after 6 weeks or so, all those cells have been sloughed off, and you got to do another treatment. Trying to do long-term gene therapy is not easy. Bone marrow transplant might be able to get at it for blood cells, but for other tissues, you just really can't get at it. So, the idea of creating genetically modified individuals is a neat one, but until you get to a spot where you can take a single cell, modify it, and then begin to propagate it, it's hard to do. I mean, saying that is I know how to do that, is you take egg or sperm and genetically modify it, and then you fuse them. But once you got a dude and you say, oh, I want him to have some sort of genetic trait now that we need to add, you can't really do it. Plants, though, we can do it. That's why we have genetically modified crops all around us, because you can take a single plant cell, modify its DNA, and actually grow back an entire plant. It'd be the equivalent of taking a liver cell from me and putting it into some kind of magic juice and have it turn into another me. There's a Star Trek episode where they do that, but for you and I, no, we're not anywhere close to that. But single cells, they can do it. All you got to do is put the DNA in there. So we have our central dogma of genetics that we want to just throw out. that DNA polymerase is responsible for replicating DNA. DNA production, that is replication. Then DNA directs the production of a portion of itself, and that is transcription. So transcription requires the enzyme RNA polymerase, but this is how we define a gene. A gene is a piece of the genome, or a piece of the DNA, that is transcribed, turned into an RNA. RNA then, of course, is translated, and we take RNA, and using the genetic code, we build up a protein. So this is essentially a non-changeable way that information is being passed around in living organisms on this here planet. And when we think about what does this mean for you, there's a lot that we can drive from this. With bacteria, we've got a whole range of different proteins that different bacteria produce that make them either non-pathogenic or potentially really, really nasty. Like the bacterium that causes botulism, Clostridium botulinum, produces the botulism toxin that is one of the most toxic molecules around. A single protein can completely block protein translation in a cell. And so you inject a little bit of Botox and you can paralyze specific cells. So we've got the cell's ability to replicate itself, transcribe, and then translate proteins. And just as thinking about some of the things we're talking about here, you have endotoxins. This is sort of the candy coating in a gram-negative cell. This is a pretty serious problem in that if you've got a sufficient bacteria population in you, even though you're taking an antibiotic, you still are going to be subject to the effects of the endotoxin. These do all kinds of things in that they are affecting the immune system, and so you can have this sort of immunological storm going on. This is what generates what's known as toxic shock. When you have sufficient bacteria load that the endotoxins are actually causing pretty serious damage. Exotoxins are the term we use for things that are soluble and they're being dumped out and released. Not all bacteria produce them, but a lot do. And these can do all kinds of stuff, like blocking enzymes. Speaking of the chloride transporter, you block the chloride transporter in your large intestine, and you have, this is what cholera does, and you have absolute massive rampant diarrhea. Oh boy. You can have paralysis, tetanus, which is tetany, is this real sharp muscle contraction. This is caused by specific bacterial toxins being released. So yeah, we could generate some really nasty bioterrorism type poisons by growing up some of these bacteria and purifying the proteins. Let's not do that. We then have a whole range of proteins and things that are producing the different sugars, and these sugars, the capsule, they can cause the patient's and the infected person's immune system to not recognize it, kind of a stealth system. The pilus proteins are responsible for, sort of the necrotic fate of different cell types, and things like methicillin-resistant Staph aureus manifests itself as first a boil that doesn't really heal, and what you got is the bacteria starting to hang there and beginning to generate a immune response that results in infection and necrosis. And because it is resistant to antibiotics, you can't get rid of it without, physically scooping that part of your body out. Oh, let's not do that. We then also have a range of different kinds of enzymes that bacteria can generate that can break apart different things. Things like proteins, the different sugars and things in this. So we have bacteria that are getting into the lining of our of our stomach, and start destroying the membrane and the sugar coating of your stomach, and you start generating an ulcer. Lastly, then, we have genes that will not actually do anything to you, but what they're doing is giving the bacteria the ability to be resistant to specific antibiotics. Now, antibiotic resistance has been around forever, but you only begin seeing it when you use antibiotics. And so the nature of bacteria is to always figure out ways to grow. And so you have an antibiotic, and it's pretty much a guarantee, just because of the numbers of trillions of bacteria that you're dealing with. One of those dudes is going to figure out a way, mutate a protein, or mutate a gene, to generate a protein that is going to be resistant to the antibiotic or is able to break the antibiotic down. I mean, antibiotics are naturally occurring products, pretty much all of them that we'll be talking about, at least in that case. And so they can be broken down. And when they are, they're no longer functional. So it is always a race between antibiotics and antibiotic resistance. And this comes from mutation. So mutation is a term we use for any change in the sequence of the DNA, of the nucleotides. And so we are all carrying different gene sequences in a few spots, and that's what gives rise to all the differences we see within the population. So these are mutations, and one of the terms with this is, how do you decide what is the baseline sequence, what is the sequence? And then changes from that are what you call mutations. And it's just a term that we use. If you're still able to get up and move around and reproduce, then whatever you've got, those mutations are non-deleterious. They may give you naturally curly hair, they may give you different colored eyes, but they're not having an effect on your ability to reproduce, and so by definition, you're successful. Go forth and have more children to spread your mutation around. You then can have mutations that actually can have some kind of damage, and happily, they're pretty rare. So we've got spontaneous mutations, and this happens because the system for replicating our DNA ain't perfect. I mean, it's pretty darn good, and we've got all kinds of error checking, but it's one of the things that if you stand around on the planet long enough and divide your cells long enough, you pretty much are going to have a mistake occurring eventually that will result in some kind of cancer, just some kind of abnormal cell division. A good immune system can pick that up and pull it out. In fact, it's estimated that just because we're standing around in sunlight, which is slightly mutagenic, just the way life is, we generate a mutation that would result in a tumor around 1 time per month. Happily, our immune system is attuned to those kinds of changes and pulls those cells out of circulation right away. We know this because people that are immune compromised begin to show particularly rare cancers. Folks that have HIV, the virus that causes the acquired immune deficiency syndrome, AIDS, they begin to show very specific skin cancers called Kaposi sarcoma that prior to HIV being spread, the CDC had seen a single case of Kaposi sarcoma. Once HIV started infecting the population, they started seeing Kaposi sarcoma all over the place. This is an example of a kind of tumor that our immune system, man, you pull that out, no problem. It is just like that mutation is something that our immune cells can recognize, see, and pull out. So we generate mutations. Mutations happen. And they are either just naturally occurring, or they can be enhanced by mutagens. And so when you have a mutagen, this is what we go from, rather than being spontaneous, to being an induced mutation. Spontaneous mutations just happen because DNA polymerase, while it is pretty darn good at error checking, is not perfect. But then standing around out in the sunlight, exposing yourself to chemicals, that kind of stuff, these are mutagens. Now, we have this idea here of a mutagen versus a carcinogen. And it's an interesting distinction. Pretty much anything that is a mutagen has the possibility of being a carcinogen. That's just the way it is. In that, if you pick up a mutation in the wrong spot, you can generate a tumor. Now, it probably takes multiple hits in a couple of different spots in the genome to actually get a full-blown metastatic tumor. But anything that is affecting your DNA has the possibility of doing that. Consequently, we take materials that are mutagenic pretty darn seriously. Now, just that said, there are a lot of naturally occurring mutagens, and there is a test that looks at the relative mutagenicity of different chemicals, and coffee can be a mutagen. Excuse me. Apparently, raspberry seeds can be quite mutagenic. But we have co-evolved with these kinds of chemicals ever since animals have been around. And so these kinds of mutagens are not particularly deleterious to us. So just because one organism is mutated by it doesn't mean that everybody's going to be bothered by it, because you may have a way of detoxifying that. But what you can say is that if something is mutagenic to an organism, it is possibly carcinogenic. And I'm trying to think about the other side of this. Not all carcinogens are mutagens. There are things that that are produced like in coal tar extracts. There's compounds that are known as 4-ball esters, and I keep on thinking I need to actually go back and looking at some of those. There are some plants that produce these things, and these actually can be very not mutagenic, but they can start to cause cells to begin dividing out of control. But pretty much, if you are a carcinogen, almost all carcinogens are mutagens, and all mutagens we have to define as a carcinogen. All right, enough of that. So we can think about mutations in a couple of different ways. A silent mutation is a mutation that changes a nucleotide, but either it's not in a portion of the genome that codes for a protein. That happens all the time. You can have a mutation that changes a nucleotide, but because of the genetic code, it may not change the amino acid that's in the protein. No big deal there either. You can have a mutation that will change an amino acid, but change it from one amino acid type to another similar amino acid type. So you have a nonpolar amino acid change to another nonpolar amino acid, what's called conservative substitution. No big deal there either. Occasionally, and it's actually fairly rare, that you can have a mutation that results in a slightly different way that protein works. And so, if you think about the number of traits within our population, while it's fairly significant, the big stuff doesn't change. I mean, if you start monkeying around with the way your face is forming, you probably don't make it out of the embryogenesis stage. So we've got a variety of different kinds of genes in the population that give rise to different traits, but you can't start monkeying around with stuff too much. And so new traits are fairly rare. And what this is the gene has been altered and it's going to begin doing something else. So we've got some things that we can think of that in mechanical ways of doing this. We can have a mutation that occurs in one generation during replication, during DNA replication, and that is going to mean a permanent change in the next generation. And most of the time, the change is not going to be significant. One of the points, and actually we can relate here, is that you and I have got about 20,000 genes, but we've got 3 billion nucleotides. We've got a bunch of DNA in our genome that isn't involved specifically in coding for proteins. And so you can pick up a fair amount of mutation and not have it affect you. If your genome is more compact, every mutation is more likely going to affect some protein. And the more compact your genome is, the more likely a mutation is to have an effect on your proteins. So we have a really big genome. Bacteria have significantly smaller genomes, and they've got their genes packed way tighter. Now, you know who has their genes packed even tighter is our little virus friends. Viruses are phenomenally compact little guys. They don't carry around a lot of nucleic acid. Consequently, any mutation in a viral genome is just about guaranteed to have an effect on one of the proteins. That's why when you have viral spread, the chance of generating new strains is almost not will it happen, but is a guarantee it's going to happen. It's going to be when will it happen. And so viral spread, and the more you replicate it, guarantee you're making mutations that will result in new strains. I mean, with a larger organism, you have mutations, and it may result in, quote, a new strain, but the only way you'd be able to find it is to actually be able to do DNA sequencing of the organism, and you find some little spot that, instead of having a T there, they got an A. Big deal. But with the virus, you're making mutations. Very likely you're going to be changing individual proteins. Oftentimes, you could have a mutation that doesn't affect the virus or causes the virus to be non-functional, but the way this system works is that mutations that result in greater infection, they start to infect more, and you start to have a larger population of those viruses. We've already seen a shift in the strain of COVID-19, and from the very beginning to now. And then the concern, of course, is that there are new strains coming along. Some may be more contagious than others. So the best way to stop these new strains from forming is to stop the transmission of it as fast as you can, because new strains just will form. So as an example, something maybe you're familiar with is the genetic predisposition, the genetic disease known as sickle cell anemia. Is a change in the protein that binds oxygen, the hemoglobin protein. And it results in it comes from a single nucleotide difference. So the normal hemoglobin has an RNA that produces, or that has a sequence of GAA. The mutation has the sequence GUA. And that difference is going to put in a single amino acid different. You're going to have a glutamine in a normal hemoglobin, but a valine in the sickle cell. That single amino acid causes a slight change in shape of the protein, and it makes it so it begins to change the overall shape of the red blood cell. So happily, most mutations don't end up having this kind of deleterious effect with a single nucleotide change. But all the ones that you know of, cystic fibrosis, muscular dystrophy, sickle cell anemia, these are genetic mutations that are typically resulting from a single nucleotide change. Yeah. So if we think about what's causing mutations, we have mutagens, but there is random mutagenesis that occurs. And so the idea is that resistance is there, but because you don't select for it, you don't see it. So within a population of bacteria or people, you're going to have individuals that are resistant to a specific kind of phenomenon, a specific drug or whatever. But until you select for it, you never see those guys come out of the population. They're just a member of the crowd. I mean, you can imagine, thankfully, if this were a zombie apocalypse, we're going to have individuals that are going to be insensitive to the disease. A really good disease is one that wouldn't do that, but let's say we have some kind of virus or something. And so it's within the population, and probably with COVID it is that way. There are individuals that are not going to be able to take up the virus in any specific way in a high level. They're resistant to it. They're here, they've been around with us all the time, but because they just are mixed in with the population, nothing special about them, it's only when you expose them to this selection that everybody else dies and they're the ones going, hey, I'm still here. And they begin to repopulate the earth. So the same thing is happening with bacteria, is that resistance is around, but only when you apply the selective pressure of an antibiotic do you see those resistant strains starting to shift the population, and everybody begins more like them. So if the mutations are pre-existing, you'd see them on a plate that was exposed to the antibiotic and not in the antibiotic. And so the pattern you'd see would be the same. You can do this by replica plating. If the mutation is induced, then you're going to see colonies that are coming up over time, randomly located, and these are things that aren't present prior to the exposure. And different kinds of chemicals are going to do different kinds of things. So it's a way of thinking about how we can have different driving forces behind developing these resistance. So the spontaneous mutations are present, and antibiotic exposure is simply revealing them. On the other hand, we can have environmental challenges that are causing mutations, and antibiotics can cause bacteria to become drug resistant. So we have these two different kinds of ways, but when we begin looking at the models, this kind of model is never seen. This is like the hypothesis. And what we can conclude from this experiment is that all antibiotic resistance is present in the bacterial population. Now, mutagenizing the bacteria is going to create more changes in things, but it's not like the antibiotic itself is causing the mutation. So this sort of model, we really just don't see this. This is the way it actually happens. So once a gene or a bacterium is resistant to antibiotic, it can transfer that resistance to other bacteria. And a couple of things that happen is that we can have movement of DNA. And transposition is a term we use for movement of genes within a cell. And this is a phenomenon known as transposition using a piece of DNA that is known as a transposon. And we've got transposons that were present in our genomes, and they are no longer functioning. Partly transposons make your genome somewhat unstable. There are a lot of insects that still have active transposons. There are a lot of plants that have active transposons. We don't. And that's partly because we're able to, we figured out a way to inactivate that, and that's how we're able to operate. So you've got the transposase. That's the enzyme that can loop out this piece of DNA, pull it out, and put it someplace else. So we can begin to move genes around within a cell, but then we can also move genes between cells. So we know that we can produce new cells through standard mitosis, and that's what we think of as vertical or binary fission. And we've seen this. So the chromosome replicates, the cell separates, and we then have two cells. So that is a standard way of transferring DNA from one bacteria to another. What is new is horizontal gene transfer. And we got a couple of ways of doing this. Transformation, I'm about to do transformation upstairs in a little bit. That's where you take cells, you have to render them what's called competent, but you hit them with a bit of calcium chloride, and you just throw DNA onto them, and they will take the DNA up. That's transformation. We also can move DNA around by infecting them with viruses. And when you're using a virus, not just naked DNA, that is transduction. Then, when we've got bacteria, they can actually produce a little tunnel or tube of cytoplasmic continuity using some of the pili, the sex pili, and they actually can transfer DNA from one individual cell to another. And this is called conjugation. And it's not mating exactly, but it's similar to, it is a way of transferring DNA. And so this is a way of transferring DNA between two cells. So these terms are how we can move DNA from one bacterium to another, horizontal transformation. And there are some specific kinds of factors that allow for horizontal gene transfer. So let's just kind of look at this here. Vertical gene transfer, we've got some kind of virulence factor. We can track it, and we can create new strains by mutation, but it is just spreading this specific gene by binary fission. But we can also have the DNA being released from this bacterium, being popped open. And through transformation, just naked DNA, it's taken up by cells and copied. And you can generate a whole range of new combinations this way. So that's one way we can think about the inheritance of vertical versus horizontal. The horizontal is going to have a mix of a range of different combinations between individuals. Not everybody is sort of getting the same thing. So the three different kinds of horizontal transfer, and this is a question you'll see on the exam a good bit. Bacterial transformation, the donor cell is releasing the DNA, or in my case, I shoot naked DNA from a tube into the recipient cells. And if you remember back, we were looking at the Avery experiment with the little dead mouse. This is what they were doing, is the roughened non-pathogenic cells were picking up DNA from the smooth cells, the virulent cells, and becoming virulent. And so this is the transforming factor, DNA. It's being picked up by the recipient cell. So something like, a antibiotic resistance gene. And in fact, what I do is I have recombinant DNAs that are coupled to an antibiotic resistance gene, specifically for ampicillin. And so I look and grow bacteria on a plate containing ampicillin. If they have taken up my new piece of DNA, they're resistant to ampicillin. Bacterial transduction is where we use a bacteria virus. And you may or may not know, bacteria have viruses just the same way that eukaryotes do. Rather than called bacteria viruses, they're referred to as bacteriophage. And a phage can infect a cell when the phage, in some cases, can pick up a bit of DNA. The phage do what phage always do, which is rebuild themselves, pop open the cell, and infect a neighboring cell. And this is the bacterial transduction mechanism. And it's just a term, but it is a different way of moving DNA around. And then conjugation is this complete other thing, in which you've got two bacteria that actually build a cytoplasmic tube between each other and begin transferring DNA that way. And so we've got a whole range of different mechanisms by which we can bring DNA from one cell to another. So what we can do is go back to this experiment and thinking about, I was saying Avery, this is Griffith? I thought it was Avery. Avery must have done some of this. I wonder if he did the next step. Regardless, we've got this experiment on which we're looking for the transforming factor. And if you remember, we had a virulent cell type and a non-virulent cell type. The non-virulent cell type was known as the R cell type, and that's because they didn't have the capsule, and so they had this roughened appearance. The smooth had this candy coating that gave them the smooth appearance. And we've got then a couple of different kinds of experimental systems here. Boiled smooth cells, they had the capsule, but they can't reproduce. The mouse is probably getting a bit of an infection or a bit of an immune response, but he's not dead. We've got the living cells with the deadly capsule. They go into the mouse, they reproduce inside the mouse, they kill the mouse. The roughened cells are harmless, live, and they're harmless dead too. You inject them, the mouse doesn't care. What was discovered then, the transforming principle, was taking the heat-killed pathogenic bacteria and the live non-pathogenic bacteria, and what we got is transformation. We've got the ability of the non-lethal bacterium is picking up DNA from the dead lethal bacterium. And we inject it, mice die, and then the cells we pull out, the bacterial infection, has the capsule. And so we know from the mechanics of this, when you take the killer cells and you boil them up, you're breaking them apart. Parts of the DNA are going into solution, and the non-killer cells are picking up a bit of that DNA, and recombination, the ability to put DNA into the genome, put it into the genome, and now it's functioning, and it can produce its own smooth capsule. And so you've got the rough cell is now been turned into the pathogenic capsule-containing smooth cell. So this is how we were able to really know that DNA is, in fact, the transforming factor. The last moment then. Transduction is looking at the way the virus works, and that the viral genome, and we'll talk about bacteriophage a little bit later, the viral genome is injected into the bacterium, and some of the viruses, when they package themselves back up, actually have a bit of the host DNA. The bacteria is lysed, the new viruses are dumped out into the media, and they infect the next guy. And they're able to, through recombination, modify the genome of the organism they got put into. And you have now a virally transmitted new gene. And we're going to stop here and talk about the next kind of mechanism of conjugation on Monday. So, it's one of the things we're going to be talking about virology here in a bit, and using bacteria and bacteria viruses as a model system works really well, because we can understand and interpret and begin imagining all kinds of viral scenarios. And the fact is, viruses can pick up DNA from their hosts. And so, in the human population, we don't really have too many viruses that are bringing pieces of mutated DNA into our genomes and causing some kind of harm. But like if you have a cat, you know that you need to have your cat vaccinated against the feline leukemia virus, because there is a virus that if the cat catches it, they get leukemia. And fortunately, like human papillomavirus is close, but not quite. We don't have any what are known as transforming viruses. Viruses that if you catch them, you get cancer. And lots of other animals, chickens, I don't think dogs have any, but mice, all kinds of animals, have cancer-causing viruses. It's one of the things I think about, if you want to make a really scary sort of observation. Imagine if COVID, rather than giving people the flu and flu-like symptoms, and yes, you can have lung problems, what if it gave people leukemia? Whew, you talk about suck. And that's a fairly rare thing, and hopefully it won't happen, but it's something to think about. All right, we're going to head upstairs here. If you want to look at plates and stuff, we can look, and that's all I know.